This is picking up on our second part of Chapter 5, covering bacteria. We pick up right where we left off, so hopefully this will work for you. Thank you. Just to follow up, again, the glycocalyx provides protection against desiccation. Also, because it has that layer there, it's a little harder to kill some of these with chemical compounds. And it protects against variations in the environment. Big thing, it provides adhesion properties for some bacteria. Now, many bacteria also have the ability to move around by using flagellum or flagella. Some microorganisms have a single flagella. Many have more than one. And what we see here is a bacterial cell with multiple flagella. But these structures allow the cell to move. These basically twist in one direction, resulting in movement forward. When the cell stops after a moment or two, it will reverse the movement of the flagella, and at that point it changes direction, and that's sometimes referred to as a tumble. So moving forward is referred to as a run. This sort of tumble that it does, and then it can change direction and go in a different way. But these allow the organism to respond to their environment. Other surface structures that exist on bacteria include a pilus, or pili, and these are like hairs. So, pili means hair. It is a hollow tube. And one thing that pili can do is allow exchange of genetic information between one bacteria and another. They can actually come up to another bacteria, lock into that bacteria's cell membrane, and copies of genetic material, including structures called plasmids, can be transferred from one cell to another, often giving important information to a cell, maybe resistance. It also functions as an attachment factor for some bacteria, so it aids them again in binding to some of our tissues. This image actually shows us E. coli bacterium with pili sticking off of it. Now, those are the shorter structures protruding, which would allow it either to communicate genetic information from itself to other microorganisms and have binding properties. Once we look inside the cell, we have some cytoplasmic structures. Now, not as complex as the eukaryotic cells. We've already talked about all of their organelles. But we have a zone where the genetic material is, and it's just referred to as nucleoid. So instead of a nucleus, we have this nucleoid located, a location of the bacterial DNA. What we see is generally a single double-stranded circular chromosome. It doesn't have proteins on it or other things that bind it up in the same way that eukaryotic DNA is. So it's much simpler. There are plasmids, which are little loops of DNA, small circular ones. They are not part of the regular chromosome, and they don't have anything to do with maintenance of the cell. They can provide resistance to the microorganisms, such as resistance to antibiotics, and this can be transferred via this pilus to other microorganisms. These plasmids replicate independently from the DNA. We also, of course, see ribosomes. Every cell has protein production, and you have to have ribosomes for that. You also have to have transfer RNA to carry the amino acids necessary to make the protein. And then we have soluble cytoplasmic contents, which are going to include the amino acids, sugars, the electrolytes, other substances that the cell needs to survive. Some bacteria have the capacity to make an endospore. That is a structure that contains the DNA of the cell, and it makes these when the environment is getting nasty enough that the cell may not survive. This is really typical in genuses of Bacillus and Clostridium, and these microorganisms can be pathogenic. What's going to happen is if the environment becomes unable to support the cell, it will make this bit of DNA, package it up tightly, and this is heat resistant. Fact is that we have to autoclave to ensure that we have killed this stuff off. Now, not only are spores heat resistant, as I said, it requires steam and pressure to destroy, but they are environmentally resistant. In fact, they can stay dormant for thousands of years. They have found bacterial endospores and brought them back to life in the pyramids. One of these is Bacillus anthracis, anthrax, which can be used as an agent of bioterrorism. You can cause these things to form spores, and then you can use the spores to infect people's respiratory tracts or wounds. This graphic just shows the basic formation of a bacterial spore, and what we get happening is a living cell. The environment changes to the point where the cell is threatened. It makes a second strand of DNA through copying what it has, and instead of dividing through fusion or fission like they normally do, it stores that and forms a spore coat, which can later be rejuvenated in the appropriate conditions. Now, I referred to fission earlier, and that is part of bacterial reproduction. Binary fission. And what happens there is instead of going through the processes of mitosis that a eukaryotic cell does, cell reproduction is a lot simpler. And what happens is that there is a copy of the DNA, so the DNA is split. DNA goes to one side of the cell, DNA goes to the other side of the cell, and it splits in two. Because it's a simple process, cells can reproduce very rapidly in optimum conditions. In fact, E. coli can double every 20 minutes. And if that were allowed to happen, if we had 100 E. coli and they doubled in 20 minutes to 200, and then by 40 minutes we had 400, and by an hour we had 800, well, if that continued, then in just 36 hours, the Earth's surface would be covered in a foot-thick layer of E. coli. The reason that doesn't happen is that, like all organisms, there are certain growth requirements. There has to be the appropriate amount of food, and there have to be the appropriate nutrients and other environmental factors, and the microorganisms simply don't reproduce at an uncontrolled rate for very long. They run out of resources. And this graphic just shows us the steps involved in bacterial reproduction. As you can see, binary fission, which is just the splitting of the microorganism into two after we have duplicated the DNA. Now, we can grow bacteria ourselves on certain bacterial growth media used in labs. There are selective media that will support the growth of some microorganisms and not others. And there are a variety of media. There are broths, which is a number of liquids that can contain nutrients, simple broth, like beef broth, in fact. Agar, which is a gelatin that's derived from seaweed. Just pure agar by itself isn't going to support growth, but nutrient agar, you supply the necessary nutrients in an enriched medium, and you're going to be able to grow microorganisms. Selective media, there are some microorganisms that grow in certain environments that other microorganisms can't. So you can actually develop a media that will grow certain things which may be used to develop bioterrorist weapons. It is a complicated process to develop some of these bioterrorist weapons, but still, it can be done with the appropriate organism and the appropriate media. Now, we've been talking about general characteristics of bacteria. Let's talk about the different types. There is archaea, and these are not viewed as true bacteria because of several structural and chemical differences in the way that the cell wall is put together and certain things in their RNA. But within that group is a class of organisms that we refer to as extremophiles. And these are ones that live in some very extreme environments. And a couple of examples. We have the thermoacidophile, which lives in very acid conditions that are also quite hot, such as hot springs. Sulfolabus acidocalderis, which grows at a pH of 1. That's getting real close to stomach pH, and it grows at 85 degrees centigrade, which is just a little bit below boiling temperature. Pyrolobus fumare, which grows 150 feet below the ocean, so it's under extreme pressure, grows between 90 degrees Celsius and 113 degrees Celsius, which is actually above the boiling point, but because we have it under pressure, that water doesn't boil. This is an example of one of the habitats for an extremophile. This is an alkaline spring in Yellowstone, which shows a form of cyanobacteria lying on the surface of the ground in front of it. There are several other archaea, and again, archaea just do some odd things. So we have methanogens, which produce methane, and they only require carbon dioxide, nitrogen, and water. They don't require light. They break down some organic products. They live in marshes and swamps and make swamp gas. Methane, which is a burnable product, so actually like a natural gas. There are extreme halophiles. These are organisms that like to live in salty waters, such as in the Great Salt Lake. We also see photosynthetic bacteria, which are not part of the archaea. This is a different group. Photosynthetic bacteria include the cyanobacteria, which is a major form. They're autotrophic, that is, they produce their own food. And to be autotrophic, you need to have chlorophyll. These can fix nitrogen. So there is some of them can fix nitrogen, which gives a symbiotic relationship with other cyanobacteria. Fixing nitrogen. Nitrogen is a necessary nutrient for the growth, and it's hard just to pull that out of the air. So some of these cyanobacteria are capable of that. Cyanobacteria, and we may have mentioned before, they were largely responsible for introducing an oxygen atmosphere to this planet. So, photosynthetic bacteria, that's the cyanobacteria. It's a major form, autotrophic, contains chlorophyll, fixes nitrogen, symbiotic lifestyle with other cyanobacteria, highly independent organisms. They don't really require anything from other organisms. They can adapt very well. Like we said, probably the first to produce oxygen in Earth's atmosphere. Here is a view of a mat of cyanobacteria. We need to stop again on the second part of the presentation. We will pick up right here and finish up with part 3. Thank you.